Hi, this is Professor Kamble here. Today we are going to discuss about next topic of our syllabus, that is different techniques in plant tissue culture. In the last lecture, we have gone through the scope, importance, and overall overlook of our syllabus. This is one of the important point of our syllabus with respect to final theory examination, in which we are going to discuss about what are the different techniques in plant tissue culture, in which organ culture is there, embryo culture is there, cell suspension culture, callus culture, anther culture, pollen culture, and their applications. Okay, so moving on to the next slide. First of all, important terminologies are there, in which first is the totipotency. It is an inherent potentiality of a plant cell to give rise to a whole plant is described as a cellular totipotency. Okay, so this totipotency is the base of plant tissue culture. Okay, if we are taking any cell of the plant, tissue, or any organ of the plant, and if we use it as an explant on nutrient medium. Okay, so before that, explant is nothing but the any plant part which is used to start the culture or initiate the culture, that is your explant. If we are using shoot tip, then shoot tip is the explant. If we are using nodal explant, nodal part, then that nodal part will be the explant. Like that, different explant can be used as a different parts of the plant can be used as explant to start the culture. And all these plant cells and tissues are having cells basically. Okay, and the cells are having ability to grow into a full plant, that is your totipotency. Okay, and because of this totipotency only the plant tissue culture got emerged. If there is a no no totipotency, there is a no tissue culture. Okay, so this inherent potentiality of the plant cell to rise into whole plant is totipotency. Second important terminology, that is differentiation. It is defined as the process by which meristematic cells are converted into two or more type of cells, tissues, or organs which are qualitatively different from each other. Okay, so differentiation is nothing but the change in the form when we are growing it onto the nutrient medium supplemented with hormones and amino acids which are necessary for this differentiation. Then de-differentiation. The term is used to denote the process of formation of unorganized tissues from the highly organized tissues. Okay, highly organized tissues are nothing but the parts of the plant body. Okay, which are having in particular function and specific tissues, from which if we are getting an unorganized tissues, that is callus. Okay, callus is one of the important example of unorganized tissue. This process is called as de-differentiation. Then redifferentiation, the process of differentiation occurring in an undifferentiated tissues. Okay, organogenesis. What is this organogenesis? As we know, genesis is nothing but the starting of that particular process or a developing process. Okay, and here it is organogenesis, that is formation of organs. In plant tissue culture, organogenesis means genesis of organs like shoots, roots, leaves, and flowers. Morphogenesis, attainment of biological organization or form, is termed as a morphogenesis. Okay, so basically in callus culture we are having unorganized tissues. Okay, and from which we are looking for the regeneration into full plantlets, and this whole process is nothing but the morphogenesis, in which different organizations or different organs got formed, that is nothing but the morphogenesis. Now here, types of culture. Okay, Now, types of culture, actually, these are not the types of culture. These are the techniques which are used in the tissue culture, and which can also be called as types of cultures on the basis of explant used. Okay, and as we discussed, explant is any part of the plant which is used to start and culture or initiate culture is nothing but the explant. Okay, in case of banana we are using internal part of the suckers. So in case of banana it is an explant. In case of sugarcane we are using upper or deeper portion of the plant. So in sugarcane this particular upper region is explant. Okay, as per the plant's changes, explant also changes. Basically explant is containing meristematic regions. Okay, from where we'll get the multiplication, or this particular meristematic tissues are having ability to grow faster, so that we will get the proper regeneration, multiplication. So first of all, embryo culture is there, in which we are culturing mature and immature embryos. Okay, embryo is nothing but the seed when it is in mature form. So sometime we are using immature embryos also. Then seed culture, in some cases it is used. Meristeme culture. Meristeme culture is again one of the important technique involved in plant tissue culture, in which only the meristematic region of the plant can be used for the culturing. Okay, in which very much, very means very less means some microns. Okay, it may be 5 to 10 microns size of the upper tip region of the plant is mainly used, which is containing the meristematic highly dividing, or the cell division rate is very much higher in this meristematic region, which is used to develop virus free plants. And one of the important reason behind using this meristematic region is it containing higher levels of auxins and cytokinins, and also growth rate is very high. And in this particular environment virus is unable to multiply, even it's unable to survive in this meristematic region, so that this meristem culture is there for the production of virus free plants, which is the one of the motive of these plant tissue culture techniques nowadays, which are used for basically plants which are free from the any type of viruses. Then protoplast culture is there. As we know, protoplast is nothing but the cell without cell wall, which is also having some important aspects. Then organ culture is there, bird culture, callus culture, and cell suspension culture. Okay, cell suspension is nothing but the uh, unorganized mass of the tissues which is kept suspending in liquid medium. Okay, in detail we'll see later. And callus culture, as we know, callus is the unorganized mass of tissue which is getting produced or nearby the explant on nutrient medium is called as callus, and the culture is callus culture. Okay, types of the culture: organ culture, culturing isolated organs on tissues such as root, stem, or leaf in an artificial medium under controlled conditions are known as organ culture. Depending on the type of organs or tissues used for establishing the culture, organ culture are named accordingly, as we saw in our previous slide. The following are the various types of organ culture and its specific purpose, in which first is the seed culture, increasing the efficiency of germination of seed that are difficult to germinate in vivo, precocious germination by application of plant growth regulators, and production of clean seedling for explants or meristem culture. Okay, so such type of the seeds are also there, crops are there in which there is a problem of germination, or some specific type of the seeds are there in which we do not have explants. So in these cases seed culture is mainly used. Secondly, embryo culture, overcoming embryo abortion due to incompatibility barriers, overcoming seed dormancy and self-sterility of seeds, and embryo risk in distant hybridization where endosperm development is poor, and shortening of breeding cycle, etc. Okay, embryo culture is mainly used for this embryo risk technique. Okay, embryo culture, in embryo culture what we supposed to do that we are isolating the embryo from the seed, and this embryo is used as an explant, and which is uh, inoculated on the nutrient medium. And why we are using this embryo culture? Because in normal crossing or hybridization, there is an cross between related species, okay, which are from same genus or which are from same family, okay. But sometimes there is an interspecific and intergeneric crosses, okay. In these cases, cross, we are able to do the cross, okay. But later on, when the development of the plantlet is there, seed formation is there, at that time embryo is unable to develop, okay, or it got partially developed, so we will not get next generation. In these cases, once the crossing completed and formation of seed takes place, we will isolate the embryo, and which will be used as explant. We will transfer it onto the nutrient medium, and we will get the full plantlets, that is embryo risk. Then dormancy, okay, seed dormancy is there, okay. As we know, seed dormancy, there is a specific period or resting period required for the seeds, required by the seeds for their next germination. That is nothing but the dormancy, and this seed dormancy can be broken by the using their embryo cultures. Okay, so in this diagram we are able to clear it out some basics of techniques used in tissue culture. So first of all, this is one of the plantlet, okay. For example, just for our understanding purpose, it may it is just consider this is a sugarcane plantlet, okay. So in this plantlet, this upper region of the plant is containing meristem, okay. So here we are able to see this very small area in some microns is having meristematic region, okay. So that can also be used as a explant. Then we are able to use the some shoot which is not containing the meristematic region, okay. Excluding meristematic region, some shoot part can also be used as a explant. Then nodes, okay. So here in sugarcane we are able to easily observe nodes and internodes, okay. From that particular, there is a growth region is there from where shootlets can be formed, or if we are able, if we are cutting them and growing in the nursery, that also we rise into the full plantlets. That is nothing but the nodes, that can also be used as a explant. And once we got these explants, we will culture them onto the nutrient medium, and finally we'll get the full plantlets, okay. Meristem culture, multiplication of the shoots, node culture, okay. And here direct morphogenesis, in which sometime this callus formation is takes place, okay. And this callus, from this callus we will get this adventitious shoots or somatic embryos, and which is indirect embryogenesis, through which we will get the final plantlet. And this callus can be used as an explant in the cell suspension culture, where we are able to keep these cultures for a very long time. And there is an again some different purposes of this cell suspension culture, okay. So this is flow chart of the process. Next, ovary or ovule culture. A common explant for the initiation of somatic embryogenic cultures for the production of haploid plants. Okay, now what is meant by haploid? That is also important. As we know, diploid is nothing but the two sets of the chromosome. As like that, haploid plantlets or haploid organism is containing a single set of chromosomes is called as a haploid plantlet. Okay, so here ovary and ovule culture. Okay, as we know, ovary is an sexual organ in the plant, okay, which is from the female parent. And before crossing, if we are using this ovary as an explant, then we will get this haploid plants. And this is one of the important purpose of ovary culture or ovule culture. Overcoming abortion of embryos of wide hybrids at very early stages of development due to incompatibility barriers and in vitro fertilization for the production of distant hybrids, avoiding style and stigmatic compatibility that inhibits pollen germination and pollen tube growth. Okay, so some of these problems are there in breeding programs in which there is an style and stigmatic incompatibility is there. Okay, whenever intergenetic and interspecies process are there. Okay, in normal process it is okay, it will grow normally, it will get develop into the full plantlet, no problem. But whenever interspecific and intergeneric process are there, there is an incompatibilities or some barriers are there. Because of that, in breeding there is a lot of problems. In that cases we are able to use ovary or ovule culture. Anther and microspore culture. Now this is also having one of the important aspect, that is production of haploid plants, then production of homozygous diploid lines through chromosome doubling, thus reducing the time required to produce inbreed lines and for uncovering mutations or recessive phenotypes. Okay, so these are some important points related to the anther and microspore culture. Okay, in these particular cultures we are discussing about this anther culture, ovary culture, then callus culture, okay, seed culture, embryo culture. So basically we have this idea of these organs, okay, no doubt, and these particular are used as a explant, so that the name is given to this culture. And also there is a particular specific purpose behind using these cultures in particular crop or plant. Then next, callus culture. Next, callus culture. So first of all, we'll see this diagram of the callus culture protocol, in which we are readily able to check what is meant by this callus. Okay. So callus is nothing but the unorganized mass of tissues which are produced on the nutrient medium when supplemented with the auxins. Okay, so basically here we are able to check, this is the culture of rice. Okay, so this is the petri plate in which nutrient media is there. And these are the seeds of the rice. And after the inoculation is nothing but the transfer of the seeds on this nutrient medium. And after the inoculation we will get this type of the callus. Okay, these are the unorganized mass of tissues which are produced on the nutrient medium supplemented with the appropriate amount of the auxins. Okay, so this is the callus. Okay, from these seeds we will get this type of the callus. And next step in the callus culture, that is multiplication or shooting. Okay, so this type of the calluses which will be transferred onto the nutrient medium, but here the hormones are different. Okay, in this first stage of the callus induction, auxins are there, but in the second case, in this particular nutrient medium, there is an multiple shoots from this single callus, and also callus multiplication can also be occurred. But here the major thing is that the formation of multiple shoots. Okay, so these are the multiple shoots. Next stage is there, that is these particular shoots can be isolated, separated from each other, and again transferred onto the nutrient medium, which is having hormones responsible for the root induction. And here we are able to check easily, we are able to observe the healthy rooting system which is got formed in the this rice plantlets. Okay, so this is the healthy rooting system which got formed with the use of cytokinins, and later on hardening is there. Okay, hardening is nothing but the transfer of the plantlets from in vitro conditions to the nursery conditions. Okay, where will be the and specific mixture or potting in which FYM is there, soil is there, sand is there. Okay, so this is about the protocol of the callus culture. Okay, next, callus represents an unorganized or undifferentiated mass of cell. They are generally composed of parenchymatous cells and usually undergo division when an explant is cultured in a medium supplemented with sufficient amount of auxins. It starts producing mass of cell from the surface of the explant. Okay, so from the surface of the explant, exactly the formation of callus takes place as we saw in our diagram. And this particular is composed from the parenchymatous cells, and this is the unorganized mass of cell. Okay, next, it can be maintained for very long time by intermittent subculturing to a fresh medium. The callus culture can be manipulated for different purposes by changing the hormone concentration in the media. Okay, so in the callus culture we are able to maintain this callus for very long time. Okay, simply we just need to subculture it, and subculturing means what? Once it completed around 3 weeks to 4 weeks on the same medium, and once we got some growth, then that callus can be transferred by cutting on a separate or newly freshly prepared nutrient media. Again there will get some growth. Okay, by this way, subculturing it on the time, we are able to multiply it, we are able to maintain it for very long period. Next, callus culture can be used for regeneration of plantlets, preparation of single cell or suspension culture, or for protoplast preparation. Okay, so these are the some purposes of the callus culture through which we are able to prepare a single cell culture or suspension culture. Then protoplast preparation also. Callus culture can also be used for genetic transformation studies. Okay, this is one of the important aspect of the callus culture in which genetic transformation studies as nothing but the study of the gene transformation, in which gene from one organism or plantlet can be transferred to the another organism through an gene transfer methods. Okay, then in some instances it is necessary to go through a callus phase prior to regeneration via somatic embryogenesis or organogenesis. Okay, so this is the intermediate stage for many cultures. That is also important point. That is, any plantlet regeneration is there from the explant. In between this, there is a formation of callus is there. Okay, in number of cases, callus culture are suitable for the generation of useful somaclonal variation and can be used for in vitro selection of cells and tissues variant. Okay, this somaclonal variation is again one of the important point of our syllabus that we'll cover in our next lectures. But for the now, just remember that somaclonal variation is soma, that is somatic cells. Okay, and variation in between the clones which are developed from the somatic cells is called as a somaclonal variation or somaclonal variants. And in this particular type of variation, we will get and useful variations, and such useful variations can be multiplied and used for the economical benefits and also in the crop improvement program. So this is also one of the important point related to the callus culture, as callus culture is an important role in the formation of genetic variability or somaclonal variation. Okay, so we'll stop here in this lecture. In the next lecture, we will discuss about methods of gene transformation, in which direct methods are there and indirect methods are there. Thank you.